Hi, my name is Darren Abbas. I'm a postdoc in Dr. Juan and Longacre's lab, and today I'll be talking about how wild-type mice dynamically alter their fibroblast subpopulations to heal dorsal skin radiation wounds. So radiation-induced fibrosis is a multi-billion-dollar industry. It is a known, well-documented sequela of radiation therapy, but the exact mechanism underlying the development of radiation-induced fibrosis isn't well understood. So the main purpose of our study was to help delineate the cellular mechanisms that promote the progression of radiation-induced fibrosis. We did this initially by pair biosing 3 pairs of Black 6 and GFP mice together. We then analyzed the blood samples from the mice 2 weeks after pair biosis to determine that there is proper chimerism of the blood. Then, after chimerism was confirmed, we radiated the dorsal skin of both mice with 5 grays of radiation every other day for 12 days, totaling in 30 grays. We then allowed the mice to recover for 4 weeks, and we harvested the skin samples of both the Black 6 and the GFP mouse. And as you can see in figure 1, the GFP mouse has a significant amount of green cells noted within the dorsal skin after radiation wounding, but the Black 6 mouse has essentially no green cells present within its skin after radiation exposure, which confirms our hypothesis that radiation fibrosis is a local response instead of a systemic response. We then took 30 Black 6 mice and had them undergo a similar radiation regimen as described before, except now we harvested the skin at weeks 0, 1, 2, 4, and 8 after radiation. We then analyzed these skin samples both histologically, and we sent the skin samples for single-cell RNA sequencing. So as you can see in figure 2, we performed immunofluorescence on the skin with CD31 markers to determine the amount of inflammation and angiogenesis occurring. And as you can see in the acute phase of radiation wound healing, which we described as week 0 to week 2 after radiation therapy, there's a significant increase in CD31 expression. But by the chronic phase of radiation wound healing, which is week 4 and week 8, there's a significant decrease in CD31 expression, meaning in the chronic phase of radiation wound healing, a hypovascular plane develops. We then did further histological staining with H&E to determine the dermal thickness, as you can see in figure 3. And we've determined that there's a significant progressive increase of dermal thickness throughout our experimental timeline. We also analyzed the collagen density using Mason's trichrome. And we also noted that there's a significant increase in collagen density throughout our experimental timeline. We then did picrocereus red staining under a polarized light, as you can see in figure 4A, to help determine and analyze the collagen ultrastructure. In the control mice, we noted that there is a basket weave appearance of the collagen fibers. However, by week 8 after radiation wounding, that there is a more linear progression of the collagen fibers. And using a machine learning algorithm, we then took these picrocereus red images and graphically displayed them in a UMAP, as you can see in figure 4B. And you can see that throughout the timelines, these collagen fibers tend to cluster together, whereas in week 2, as the green shape, week 4 in the blue shape, and week 8 in the pink shape as the most distinct clustering of the collagen fibers as they progress from basket weave to a linear organization. Then in figure 5, you can see we performed single-cell RNA sequencing, and we demultiplexed and used log-normalized cell counts and identified 5 transcriptionally distinct fibroblast subpopulations that we've displayed in this UMAP. And in figure 6, you can see that the distribution of fibroblasts within each of these 5 clusters throughout our experimental timeline changes, where we see a significant increase in cluster 2 at week 2, a significant increase in cluster 3 at week 8, meaning these fibroblast subpopulations dynamically alter their expression throughout the time course of radiation wound healing. We then took these 5 distinct clusters and we analyzed the top 100 genes in each of these clusters, cross-referenced them with Enricher to determine the relevant genetic pathways. And we can see that some of our clusters are analogous with previously described fibroblast subpopulations in our lab, where our cluster 0 is similar to the mechanofibro subpopulation of fibroblasts. Our cluster 1 is similar to the activated responder or innate responder fibroblasts. However, we do have a new cluster in our cluster 3, which is the transcriptional regulation fibroblasts, which has not been previously discovered. So in conclusion, in radiation wound healing, with our study, we've determined that radiation fibrosis is a local response instead of a systemic response, that we get an increase in CD31 expression and inflammation in the acute setting, but by the chronic phase, we get a hypovascular field that develops. We see a progressively increase in dermal thickness, collagen density, and collagen linear organization over time with radiation wound healing. We've identified 5 distinct fibroblast subpopulations, which have never been discovered before for radiation wound healing. And we've also discovered that these subpopulations alter their expression throughout the time course of radiation wound healing. Future directions in which we can go from this experiment forward are recreating our experiment with a rainbow mouse model to determine clonality of the fibroblast subpopulations, which we are currently performing right now. And preliminary data suggests that there is a clonal response in the acute setting, but by the chronic phase of radiation wound healing, we lose that clonality, and there's one dominant cluster that overtakes the rest by week 8. Furthermore, we can further analyze the genes within each cluster and see if there are any pertinent genes that can be modulated in order to decrease fibrosis throughout radiation wound healing. So thank you for your time and for listening today, and I'll take any questions.